Hello, and thanks for joining us today for the latest Technology Networks webinar, a microphysiological model of metastatic progression. I'm Vaish, the account manager for CM Bio, and I'm here to moderate the event. I am pleased to have Dr. Amanda Clark joining us today as your presenter. A warm welcome to you. Dr. Amanda Clark is a research assistant professor at the University of Pittsburgh. She received her PhD in biomedical research from Griffith University in Australia and relocated to the University of Pittsburgh in the United States to pursue her postdoctoral training. Dr. Clark completed her training in the laboratory of Dr. Alan Wells, working on establishing an all-human microphysiological model of dormant emergent metastatic progression. She is now a research assistant professor at the University of Pittsburgh, where she focuses on utilizing multi-organ microphysiological models to investigate how distant inflammation drives metastatic recurrence. Following the webinar, we will have a Q&A session and we welcome any questions that you may have. You can submit questions for the Q&A session at any time during the presentation. Simply enter your question into the box on the right-hand side of your screen and click send. We will answer as many as possible during the time available. For any questions we don't have time to get to, we will be sure you are contacted offline with an answer. If you experience any technical issues, please click the chat icon in the bottom right corner to request support. Please remember you can ask questions at any time, even whilst watching the webinar on demand. So without further ado, I will now hand over to Amanda.
Thank you, Vaish, for that kind introduction. And everyone for watching today. I'm going to start off by talking about metastatic disease, then give some background on how we model metastatic progression, go on to talk about using these systems to look at crosstalk in the metastatic microenvironment and how the scaffold matrices can impact metastatic progression and therapeutic efficacy. Metastatic progression is associated with poor patient prognosis and is the major cause of mortality with patients with solid tumors. Unfortunately, our advances in our abilities to remove and treat the primary disease have not yet translated into sustained success against metastases. And subsequently, metastatic disease remains largely incurable. As you can see on the right, for the 5-year relative survival, this is cumulative data for about 10 different cancers. Localized disease has a 5-year survival of around 90%, whereas distant metastasis has less than 17%. And in part, this is due to the complexity of metastatic disease compared to primary. So metastatic disease is much more complex as there are more steps involved. So cells within the primary tumor undergo an epithelial to mesenchymal transition, which enables them with motility. They then use this to enter the circulation and travel to distant organs where they extravasate and can colonize the distant organ. They can either colonize and outgrow immediately into overt disease, or they can undergo another mesenchymal to epithelial reverting transition and colonize a single or a small cluster of cells and enter a dormant state. And they lay in this way as clinically undetectable cells for extended periods of time, for years to decades before suddenly reemerging and undergoing a second epithelial to mesenchymal transition and outgrowing into overt disease. So this latter aspect of metastatic progression is a major obstacle. We don't yet fully understand why cells enter and colonize as a dormant state and why they emerge. We know that emergence is associated with inflammation, but we don't yet understand the specific drivers behind this. So part of this lack of understanding is due to an absence of tractable experimental model systems that can look at these later stages of metastasis. So to look at this, we turn to using microphysiological systems. And as no doubt you've heard from other seminars and webinars from CM Bio and this seminar series, these systems are able to both complement existing models and overcome some of their limitations. And in part, this is due to their ability to reproduce tissue level functionality and their ability to have to be viable and functioning for extended periods of time, out to at least 1 month. And when we're thinking about metastatic disease, one of the major obstacles is the lack of effective therapies, and these systems have improved preclinical predictability of drugs over the prevailing animal systems. And when we're looking at trying to model metastatic disease, one of the model organs that enables to look at both metastatic progression and look at drug efficacy is the liver. So this is because it is not only a common site for a wide range of carcinomas, but because it is also the main site of drug metabolism and limiting drug toxicity. And when we're thinking about modeling the metastatic site, it's really important that when we create this in a hepatic environment that we use both parenchymal hepatocytes and non-parenchymal cells. So for the liver, the three main non-parenchymal types are the Kupffer cells, the stellate cells, and the liver endothelial cells. And all of these cell types are important for liver functioning, optimal drug metabolism, and creating a physiologically reflective microenvironment, which is really important when we're trying to recapitulate and investigate something as complex as metastatic disease progression. So in moving forward with trying to do this, we adopted the legacy liver chip model to investigate metastatic disease progression. So using the legacy liver chip, we seed in the hepatic cells on day 0. And then on day 3, we seed in 500 fluorescently labeled breast cancer cells. And what we see is that there is a mixed population of cells that colonize. We have some that colonize and outgrow and are actively growing. And there's another subpopulation that actually colonize as dormant. So here you see 2 actively proliferating cells, and this is indicated by incorporation of EDU, and then 2 up the top here that are dormant. And this is on day 7 of culture. So this actually happens without any exogenous manipulation or genetic manipulation of the cells. And when you take this out to day 15 and look for cells that are both EDU and Ki67 negative, we have an aggressive breast cancer cell in here. About 33% remain dormant. And for the less aggressive MCF7s, approximately 45% of these cells are still dormant on day 15 of culture. And the other thing I'd really like to mention is that these cells aren't merely sitting on top of the hepatic tissue. They're actually integrating and residing alongside the hepatic tissue. So they're physiologically colonizing this 3D metastatic microenvironment. So once we had established that we had this interesting finding that there was a population of both actively proliferating and dormant cells in the hepatic niche, we wanted to see what would happen if we treated this dual population with chemotherapy. And sure enough, we saw what we see in the human patient. The actively proliferating cells were eliminated by standard chemotherapies, and the dormant cells survived. And to confirm that these weren't merely just dead or senescent cells that were sticking around in the 3D tissue, we exposed the cells to a pathophysiologically relevant stimulus of LPS EGF. So LPS is a common portal pathogen received by the liver, and EGF is produced upon injury to the liver. So we used a combination to see if we could promote the outgrowth of cells. And sure enough, we were able to see this, and we're very excited by this. So we were able to essentially mimic dormant emergent progression seen in the patient. Cells colonize as a dual population of actively growing and dormant. The actively growing can be eliminated by standard chemotherapies, and then the remaining dormant cells can be reawakened to start growing again. So once we had established that we had this model of metastatic progression, we were wondering whether we could start to probe the microenvironment to see what was going on. One of the added advantages of using the legacy liver chip system and the liver chip system in general from CM Bio is that it comes with a lot of effluent sampling. So what we did is we looked at the effluent on the 3 different cancer cell types scenarios. So the growing, the dormant, and the emergent on day 15, and compared this to the hepatic niche without tumor cells. And what we found for the dormant cells is that they were associated with a signaling profile that was reflective of cellular quiescence, even more so than the hepatic niche, tumor-free hepatic niche. Meanwhile, the actively outgrowing, so the growing and the emergent scenarios, were associated with an inflammatory signature. Interestingly, the way in which they differed was that the emergent had an upregulation of 15 different cytokines that were associated with being produced by immune and stromal cells. So this indicated to us that what might actually be going on is that the LPS EGF would be activating the non-parenchymal cells in the liver, which are then secreting cytokines and chemokines that are promoting the outgrowth of the cancer cells, rather than the stimulus directly activating the outgrowth of the dormant tumor cells within the hepatic niche. The other important finding here is that given the minimal tumor burden, so if you remember back to the start, we only seed in 500 cancer cells. This is in comparison to the 1.6 million cells of hepatocytes and non-parenchymal cells that we seed initially. This means that the markers that we're detecting likely represent changes in the microenvironment rather than the tumor cells. So when we're looking at biomarkers, most people look to see and derive signals from the tumor, but this indicates that maybe we should be looking at the microenvironment in the instance of dormant and early metastatic tumor cells. So to summarize, using this metastatic MPS model, we're able to mimic breast cancer progression as observed in human patients. Actively proliferating cells are eliminated by chemotherapy, and those surviving are dormant and are capable of being reawakened by an inflammatory stimulus. It also highlights that we're able to start using these models to elucidate candidate biomarkers of dormant and early emergent cancers, which we have not really been able to do prior. So moving on to the next part, I'd like to talk to you about how we could use these systems to look at the crosstalk that's going on in the metastatic microenvironment. So over the last decade or so, one of the key signaling elements that's been involved in cell-to-cell communication are exosomes. So in particular for the metastatic microenvironment, tumor-derived exosomes have been shown to be key promoters of tumor progression and drivers of metastatic disease. And they do this through creating a pre-metastatic niche, which promotes and enhances their colonization and survival at the metastatic site. However, what hasn't been well investigated is the role of normal tissue-derived exosomes. Very little is known about how cells within the metastatic niche affect tumor cells that are colonizing at that metastatic site. So to look at this, we first isolated exosomes from tumor cells, the same tumor cells we used in the dormant emergent progression model, and then looked at what happened to seeding. So we had the exosomes and we educated the hepatic niche for 48 hours with these tumor-derived exosomes. And what we found is that tumor-derived exosomes were able to enhance the efficiency of colonization and integration into the metastatic niche. This is consistent with findings from other labs. However, when we followed this out to 15 days, what we found was that proliferation did not follow. We assumed that if there was enhanced colonization, this would also be associated with enhanced outgrowth. However, it wasn't. And this was a surprising finding to us. And at this moment in time, we weren't necessarily sure what was driving this. And after a few more investigations, we ended up following up with the exosomes derived from the hepatocytes and MPCs. So we isolated these and then treated the breast cancer cells for 48 hours. We then looked at the mRNA of MDA231 breast cancer cells that were treated with the exosomes of hepatic niche. And what we found is that there was a large upregulation of mRNA associated with a mesenchymal to epithelial reverting transition, so more dormant phenotype, and also reduced proliferation and reduced invasion capabilities. Conversely, there was a downregulation of those associated with proliferation and the invasive phenotype and outgrowth. We then looked at the cells in culture to see what phenotype was being altered by the hepatic and MPC exosomes. And what we found is that the exosomes actually increased the expression of E-cadherin in 2 breast cancer cell lines. And E-cadherin is a prototypical marker of dormancy. So this indicated for the first time there was a role of the normal tissue in actually promoting quiescence of colonizing tumor cells. So to summarize, we're able to use these MPS models to look at exosome-mediated crosstalk. And similar to other findings, we found that tumor-derived exosomes are able to enhance their own colonization. But a novel aspect that we were able to elucidate was that the hepatic niche actually differentially regulates other parts of the metastatic niche and limits proliferation of cancer cells and promotes a more dormant phenotype over time. So if we then move on to the impact of matrix on modeling metastatic disease and also evaluating therapeutic efficacy. The standard scaffold used is a high impact polystyrene in the system. However, cancer is dramatically affected by stiffness. So stiff matrices favor cancer outgrowth and prevent dormancy. So we're wondering what would happen if we recreated metastatic progression in a softer hydrogel that more closely aligns with the physiologic mechanical properties of the liver. This is the hydrogel that we used on the left, which was supplied by Dr. Griffith's lab. And then we also have the high impact polystyrene on the right. The mechanical comparative to the liver is the polystyrene is about 3,000 gigapascals, whereas the hydrogel is about 300 kilopascals. So it more closely aligns with that of the liver. So what we did was we first wanted to know, is the hepatic tissue comparably functional in the hydrogels as with the standard polystyrene that is well established? And what we see is that we had comparable tissue formation between the polystyrene and the hydrogel. As you can see, there are less channels in the hydrogel. There's about 41 compared to 301 in the polystyrene. And we also looked at cytokine P450, four different ones on day 3 and day 13, and we saw that they were comparable over time. So now that we established that the function was comparable between the two, we wanted to see what would happen when we seed in the breast cancer cells. And what we see is the hydrogel dramatically augments the occurrence of dormancy. And this is further shown here in the hydrogel image below, where we compared 100 to 500 and then 1,000. So 10-fold times the original seeding amount. And what we see is that they still maintain this dormant phenotype. The other thing we looked at was the signaling profiles similar to that we did in the previous model. And what we see is the hydrogel is able to more closely elucidate what is going on with the crosstalk between the cancer cells with and without. The next thing we wanted to look at was the effect of chemotherapy between the two scaffolds. And what we did was we treated them with different doses of doxorubicin and cisplatin, common chemotherapies. On the left, you can see the polystyrene scaffolds, and you can see a dose response curve to the chemotherapy. And then on the right, you can see the response of the hydrogel scaffolds. And these scaffolds, you can see that there wasn't much cell death going on or elimination from the chemotherapies. And this is to be expected because the cells are predominantly dormant. What wasn't expected is that the lower doses of doxorubicin and cisplatin were associated with somewhat of an outgrowth or emergence of the dormant cancer cells. And you can see that here on the left, in the middle, and on the right for cisplatin. And this might be because the lower doses of chemotherapy may be causing stress to the responsive MPCs, which are then secreting signals to promote the emergence of dormant cells. And if this is reflective of the human situation, this has significant impacts for administering therapeutic doses. And we really want to be accurate about this, because if we're not, we may be portending metastatic related mortality by promoting the outgrowth of these dormant metastases. So to summarize, matrices that more closely align with the native physiologic and mechanical properties of the liver allowed us to improve the recapitulation of dormancy. So we'd maybe want to use this system if we're focusing on investigating dormancy more so than emergence. And it also allowed us to facilitate insights into the effect of different chemotherapeutic doses and showed a previously unsuspected behavior of dormant micrometastases as being responsive to lower doses of chemotherapy and not necessarily in the good way. So finally, I'd like to acknowledge my collaborators and colleagues at the University of Pittsburgh, especially Dr. Wells, who brought me onto this project approximately 8 years ago now. And I'd also like to thank my colleagues and collaborators at MIT, particularly Dr. Griffith and Loffenberger, for their continued advice and support over the years, and also CM Bio. Thank you, and I'll take questions.
Perfect. Thank you very much, Amanda. I'm sure that everyone will agree that there were some excellent points raised during that presentation. So we can now begin the Q&A session. I just want to remind everyone that you can submit a question to Amanda. Should you enter your question into the box on the right-hand side of your screen and then click send, and we are going to try to answer as many as possible during the time available. So we have had some already, so I will make a start. So we have one, Amanda, thank you for a nice presentation. Wondering if you investigated the role of cyclic dosing on your quiescent cells, chemotherapeutic resistant cells, and what method did you use to track the quiescent cells that re-entered the proliferation status again? Oh, I'm wondering if perhaps Amanda is on mute there. Hi,
Yes.
Amanda. Hi, Amanda.
Apologies.
Perfect. No, that's absolutely fine. Do you want me to ask the question again, or are you okay?
Yes, if you could ask it again,
Yeah,
that would be
absolutely, no problem. Okay, so the question was, did you investigate the role of cyclic dosing on your quiescent cells, chemotherapeutic resistant cells? And if you did, what method did you use to track the quiescent cells that had re-entered the proliferation status again?
So the way in which we tracked the cells entering proliferation again is we had exposed them to EDU, and we exposed them for 48 hours, and that's how we saw that they had started outgrowing again for the day 15 emergence. For the initial treatment that we showed, we just treated them for 3 days, and the doses were changed once. We have done one experiment where we took it out to 29 days. And in this scenario, we had given the chemotherapy and driven emergence of the dormant cells, then allowed them to grow until day 21 when we gave them another round of chemotherapy. And we gave them some doxorubicin, so the same chemotherapy. And in another group, we gave them cisplatin, which was a different chemotherapy. And what we found in this scenario is that the different chemotherapy was more effective than the original chemotherapy that we used, which is reflective of the human situation.
Perfect, thank you so much. So how long can this model be cultured for when studying metastatic disease, and have you run any long-term studies?
So we've taken it out to 29 days, and it maintained its viability and functionality. And we were able to do the dosing that I just discussed. So we were able to do 2 different rounds of dosing to see how the emergent cells responded to the original chemotherapy. And that worked really well for us.
Perfect, thank you. So when working with hydrogel, did you use the same MPS system as the standard scaffold used in your previous data?
Yeah, so the system itself was the same. The only thing we changed, so in the liver chip model, you have a filter and then you have your polystyrene scaffold. What we did is we just switched out that scaffold itself for a hydrogel. And because the hydrogel is quite malleable, it has the consistency similar to, say, a contact lens. We had to have that pre-attached to the filter. And so we just switched those out directly. It was relatively easy, and it was actually Dr. Griffith's group that created those for us.
Wonderful, thank you, Amanda. So are these studies under normoxic for liver, or any info on the impact of fluctuations in oxygenation on dormancy?
So we haven't yet been able to look at that. These are just the standard levels. So when this system was originally created, it was created so the liver was cultured in oxygen levels similar to that in vivo. So there's a paper way back when, in maybe 2000, from Dr. Griffith's group, showing how the oxygen levels mimic that of the liver in vivo. But we ourselves have not manipulated the oxygen levels to which the liver and the dormant cells are exposed to. But that is a great suggestion.
Great, thank you. The next one. So are metastatic tissues, less stiff than primary cancer tissues? And are there any differences between the metastatic tissue fibroblasts and primary site fibroblasts?
So I think that's a great and complex question with many elements. I'll see if I can answer them all. So with respect to the stiffness of a primary tumor versus the secondary site, that's going to really depend on the tumor type that you're looking at. And it's also in the metastatic site going to depend on the organ, so the metastatic organ that it has colonized, and also how big your tissue is and how much the tumor cell is, nodule, and how much of the tissue it has overtaken. So obviously with increasing size, it's going to become stiffer. As to the difference between fibroblasts at the primary site and the metastatic site, that's going to, obviously they're going to be different because your fibroblasts are coming from the primary site. So in breast cancer, you're going to have fibroblasts that are within the normal mammary tissue that are transitioning to become cancer associated, whereas say in the liver, you have different cells there that are transitioning. So you have more of a population of, you have like your stellate cells, which maybe more closely represent what you get typically from a fibroblast. They can become myofibroblasts. So it really, they do differ between the two sites.
Wonderful, thank you, Amanda. So how easy is it to work with exosomes in this liver on a chip model?
It's actually very easy. Exosomes can be isolated through the standard way. You can do that through either ultracentrifugation or using kits or the gradient separation. And then when you isolate them, you can either use them immediately or store them at 4 degrees for up to a week. And alternatively, you can also freeze them until you need them. In our situation, we replaced them. We added them, and then 24 hours later, we added them again. So they had a full exposure of 48 hours. But that all depends on what the question is you're asking and in relation to the exosomes using this kind of liver chip.
Perfect, that makes sense. So when you were sourcing the right liver on a chip model for your studies, what key features did you look for?
So for me, it was actually the system used by my mentor, Dr. Alan Wells, when I arrived in the lab. But if we're talking about desirable aspects maybe of what we want out of a 3D liver chip model, then I'd say cellular composition is really important. So the liver chip allows us to include both the parenchymal and non-parenchymal cells. So this is important for us to be able to faithfully recapitulate not only the physiology of the metastatic organ, but also recreate that cell-cell and signaling crosstalk between the tumor cells and the liver, which is important when you're looking at a complex disease such as metastasis. The microfluidic aspect was also very desirable because the ability to control fluid flow creates relevant shear stress forces and oxygenation, which subsequently helps extend the culture of liver cells out. So under normal 2D circumstances, they are only viable for 5 to 7 days. And then also being able to use this system for multiple readouts to look at tumor burden, look at genomic, proteomic data, metabolic data, and also look at the efficacy of therapeutics. And the other thing that it also enabled us to do for metastatic disease was modify the system in real time. So we could manipulate what was going on within the metastatic niche throughout. We could seed cells in on day 3, which is really important. So that meant that the organ tissue was able to develop properly before seeding on cancer cells, because this is what happens in vivo. Many other systems require both to be co-cultured at the same time, which really doesn't reflect what happens in vivo.
No, that's wonderful. Thank you, Amanda. That is all we have time for today, but I just want to remind everyone that for any questions that we didn't have time to get to, they will be answered offline as soon as possible, and you can continue to ask questions even whilst watching this webinar on demand. A certificate of attendance is available to download from the Handouts tab on the right-hand side of the webinar platform. This webinar will now be made available on demand, so if you do have any friends or colleagues that you think would be interested in this webinar, please do feel free to share the link with them. Thank you everyone again for watching and for taking the time, and huge thank you to Amanda for such a wonderful presentation.